everyone, and welcome to another episode of Seed Speaks. My name is Allie Roden, and I'm the digital media producer here at Seed World Group. Today, I am absolutely thrilled to be your host. I'd like to welcome you to our first episode of Season 7 of Seed Speaks. As you saw last week on our commercial, in this season, we're diving into plant breeding to help us understand the evolution our industry has undergone. This week, we're addressing the foundations of plant breeding and the crucial role Gregor Mendel played. To help us learn more, we've got three fantastic panelists joining us. John Purcell, Chief Strategy Officer at Unfold, Marcel Bruins, European Seed Editor and founder of Bruins Seed Consultancy, and Michael Mazourek, Calvin Keeney Associate Professor of Vegetable Breeding at Cornell University. Thank you so much to all of you for joining us. So let's start with some introductions. John, can you tell us some more about yourself?
Sure. Thank you, Allie. It's a pleasure to be here on the show today. So I am the Chief Strategy Officer for Unfold. We are a seed company. We'll also focus on digital solutions for the vertical farming sector. I've been in food and ag for about 35 plus years now. I've been on the vegetable side of the business for the last 12, 13. Prior to moving over to Unfold, I actually headed R&D for the vegetable division of Bayer Crop Science. So long-term Aggie, love the veggie world. And my interest is not just on the professional side, also on the personal side. I'm part owner of a family ranching operation that my brother does a great job of running full-time in Montana. So thanks, Allie, and really looking forward to the conversation today.
Awesome, thank you. And Marcel, can you tell us a little bit more about yourself?
Sure, thanks, Ali, for the invitation. I've studied plant breeding. I did my PhD in plant breeding in wheat, fusarium resistance in wheat. After my PhD, I worked for 10 years in a vegetable seed company, then moved to ISF in Switzerland. And since the past 10 years or so, I've been an independent consultant, helping out profits and non-profit organizations with all their questions related to seeds and grains and intellectual property, biotechnology, and so on.
Fantastic. And finally, Michael, can you please share some more about yourself?
Gladly. Yeah. So I'm a vegetable breeder and geneticist here at Cornell University, working on cucurbits, peppers, beans, and peas, using some of Mendel's genes. Now I'm getting ready to teach plant genetics, where we're looking at some of Mendel's discoveries and using some of my advisors' teachings, where I learned to reconceptualize some of what I had been taught in the textbooks about Mendel. I also am a co-founder of Row 7 Seeds, on the board of the Open Source Seed Initiative, and grew up with an ag background right next door, so very much trying to do what I can for the local farmers and food system in my region and share it as broadly as we can beyond.
Thank you. I'm super excited for this conversation. So let's go ahead and dive in. To start off, I wanted to give viewers a brief background of Mendel and his work and why he's known as the father of genetics. So Marcel, do you want to go ahead and kick us off?
Yeah, sure, with pleasure, Ali. And first off, really great that you guys have chosen to start your series on plant breeding with an episode of Mendel, because he really has been the father of genetics, the founder of everything that all plant breeders base their work on. But you asked me for a bit of background on Mendel, so I will have to dive into a little bit of history here. Mendel was born in 1822, so a little over 200 years ago, and he was born in a small town in Moravia, in the region of Moravia, which is now Czechia, the Czech Republic. And his family had a farm. And at primary school and high school, he excelled in natural sciences. He had a very keen mind, and he also excelled in mathematics. And but rather than continuing on the family farm, he chose to enroll in the St. Thomas Monastery in Brno, in the city of Brno, which is the second largest city in Czechia. And 2 years later, he was ordained into the priesthood there. And now a fun fact, he was born as Johann Mendel, but when he became a priest, he changed his name to Gregor Mendel. We know him all, of course, as Gregor Mendel, but in fact, he was born as Johann. And also good to remember that he was a friar. He became a friar, not a monk. Most people think Mendel was a monk, but he was a friar. And the difference is, of course, that a monk has a lot of religious responsibilities, and a friar has much more freedom and is encouraged to do a lot of teaching and do a lot of research. So that's really where Mendel's heart was lying. And he had a big dream. He really had a big dream. He wanted to become a teacher. But he also had one huge problem, that was his anxiety, his stress for the exams. Of course, also back then, when you wanted to become a teacher, you had to pass some exams, and he could not do it. He was so stressed out every time. He tried a few times, and he failed the exams. So that dream went up in smoke. He did go for 2 years to Vienna, and that was really the place where he fine-tuned and developed his research skills, his methodological skills that he would use later on in his famous research on peas. So after Vienna, he came back to Brno again, to the monastery, and that's when he started his famous crossing experiments with peas. He started off with 34 different varieties, I think we can call them, of peas, and he looked at 7 different morphological characteristics of flower color, the color of the peas, the color of the pods, the wrinkling of the peas, and so on. And all he wanted to do was see if he could create a new variety with his crossing. And that led to 8 years of crossing all those different peas in different combinations and keeping meticulous check of all the offspring that he found, ranking them and classifying them for all those morphological characteristics. And after 8 years of research and tens of thousands of plants that he studied and kept track of, he started to see a structure. He started to make sense of what he was seeing in all those numbers, notebooks full of numbers. And he was able to conduct 3 different laws on genetics out of all that work. And those are the law on segregation, the law on independent assortment, and the law on dominance. Those are the 3 most important laws that we now use in genetics. I won't go into the details of these laws, but that is really the crux of Mendel's work. And whenever traits, morphological traits in a plant or in an animal or human behave according to the rules as set up by Mendel, we call that Mendelian inheritance. And then, so he found these results. He presented his results in 1865 in 2 lectures for the Society of Natural Sciences there in Brno. And he also published his results, and I have here a copy of the original proceedings of his work from 1866 in German, of course, and he published that his result and sent it to, and that's very interesting, 133 associations of natural sciences. So he sent it all over Europe, and he sent it to the major libraries of that time in various countries, and nobody picked it up. Nobody could understand the groundbreaking results that he had found. 2 years later, Mendel became the abbot, the head of the monastery. And of course, that took him away completely from all the research, because he had a lot of other responsibilities, civic responsibility as the head of the monastery. And so during his life, his work, his findings were largely ignored. And until the spring of 1900, in the year 1900, 3 different independent botanists, they confirmed the results of Mendel. There was De Vries from the Netherlands, Correns from Germany, and Czermak from Austria. And all 3 had done independent crossing experiments and were able to confirm and say, hey, guys, what Mendel has found was really accurate. That was the way to go. And that rediscovery of his laws set everything in motion, got the ball rolling. Everybody started doing research on plants and animals and humans. And that's a bit of the background on Mendel. Ali, back to you.
That was awesome, Marcel. Thank you. Michael or John, do you guys want to add anything to that?
I think I can add. So as I'm preparing to teach Mendel in my plant genetics course this spring, there's 2 things that Marcel touched on in that fantastic summary that are going to be part of how I teach it. I learned it from my advisor, Molly John. It was how she led off her advanced plant genetics course. One is, I love that Marcel went to, he has that copy of the original paper. So he has like the true citation. There's a lot of how we view Mendel or things that have become associated with Mendel over the years, and the idea of the Mendelian trait. They're all great, but they don't really take us back to what they were conceptualizing then and how they were conceptualizing. The other one, as he touched on, is Mendel as a statistician. Great with looking at the numbers and patterns in plant breeding. We say there's a lot of people that talk about spending time with the plants, and then they'll start to talk to us. So the idea is this familiarity will eventually give you this great insight once you really can notice those details. And so Mendel and finding those contrasting character states, that was huge. There's the categorical traits, tall and short, round and wrinkled. That was a new way to approach it. And he approached it through ratios instead of percentages. So that's a key thing in how he was approaching things differently. And just also in his work, it took so long to really get integrated, as Marcel said, because it really was a paradigm shift. Up until that point, people had been looking at this blending, and it's blending of not maybe how we understand it today, but 150, 200 years ago, the concepts of what was being blended. You think about how crude medicine was in comparison then. So this idea of blending, it was kind of nebulous. But now with Mendel coming in and having a way to reduce it, have a really scientific approach to, like, there's these character states, there's something he can work with. He didn't get stuck on the exceptions. Other people are later working on the exceptions. So the idea of this paradigm shift and how it's critical to science, one of the big things to look at is that 50 years it took for Mendel's ideas to really get integrated into the chromosome theory of science and to really take off, I think. So that was a big part of it, is that paradigm shift.
Fantastic. And John, is there anything else you want to add to close out that question?
I just think, well, first of all, Marcel, great job. That was a great history lesson. I loved it. But I think the 2 elements, and Michael touched a little bit on this, though, is I think it's sort of interesting how, as we think about where breeding is going and sort of the current state and future state, the mathematical elements to it. And the fact he was classically trained or his formal training in mathematics, and his reliance on really scale. One of the things that allowed him to make some of the conclusions he made was the scale at which he did that work, which allowed for the statistical significance. And that's all very relevant to how we think about modern breeding as well. So it is interesting, we kind of know you hear the sort of the story of Mendel, but I think some of those elements on the quantification, the mathematical elements and the statistical significant elements that were really critical for his work to bring the conclusions it did are so relevant today, and actually are pretty much a strong indicator of where modern breeding technology is at this point.
Awesome. So that leads me into my next question. John, I'll kick it off with you. What is the greatest thing that you've learned from Mendel that you use in your work today, especially at Unfold?
I think the first thing that was sort of really groundbreaking is people know kind of are looking at inheritance and seeing about heritability of different characteristics, but really that laser focus that Mendel brought on specific traits, on specific characteristics. The ability to really tease apart how each of these individual traits were actually transferred from generation to generation, and using that as a means to really think about improvement is how we do it now. I mean, it's really there is a genomic aspect of this, i.e. all the different combination of traits, but in many cases it boils down to understanding what are those specific characteristics of the crop that are going to be beneficial for the crop to be successful, but then are there traits that would be deleterious to where we're trying to go as far as the productivity of that plant. So I think one of the key learnings that Mendel brought was that laser focus on specific traits that Michael laid out and Marcel laid out. But I think that plays today in how we think about really what are those characteristics that are most critical for any market you want to breed for, whether it be for open field, greenhouse, or vertical farming. Really that laser focus on what are the characteristics that are going to be most beneficial for the crop, but also characteristics that could be deleterious to the performance of that crop.
Awesome. And Michael and Marcel, take it away if you want to add anything to that.
So I say with Mendel, it's really how he approached stuff and that blending of the science with the work in the plant. So as we do our breeding work, we're combining the two for sure. I'm still working on some of Mendel's genes. He had 7 or 8, depending on the counts. We know about half of what those are. I use some of the other unknowns in my work today. And so we're still, it's just remarkable, that the 150 years since, we're still trying to discover what Mendel's genes are for half of them. We can still use them. So that's one of the fantastic things about plant breeding, is we're still able to use things while they're still in the unknown stage. I don't know exactly what the gene is, but I'm still looking at the same phenotype Mendel looked at. We have some modern tools to look at it, and it's very much a time transport for me too as I'm doing that, because as Marcel, the parallels Marcel had in his great history summary is so, and I'm also here doing it here at the university from the Czech farm, and doing it with a partner in some of the breeding work, Gregor. And so some of those parallels of what I'm doing versus what Mendel were doing, and that it's 150 years and we're still working on some of this, is present in my mind on a daily basis.
Cool. And Marcel, anything else?
Yeah, sure. Now, the greatest things that we learned from Mendel are, of course, his 3 laws. But apart from that, one of the things that I can really appreciate is that scientific rigor and perseverance eventually pays off. He worked for 8 years on these experiments. He analyzed tens of thousands of plants, and he didn't get the recognition in his lifetime. But he was sure, he had a very, very serene confidence that he had found something really groundbreaking. And just a few days before his death, he commented in 1884, and I quote, "My scientific labors have brought me a great deal of satisfaction, and I'm convinced that before long, the entire world will praise the results of these labors." So he was, as Michael said, laser focused, on the results. He knew that he had found something really groundbreaking, and it was just a matter of time before people would be able to understand what he had found. So that is one of the take-home messages of the life of Mendel for me.
Awesome. So I'm going to go ahead and take an audience question, because it kind of plays into one of my own personal questions I had prepared. But Ashley Robinson wants to know, where would plant breeding be without Mendel? And I'm going to go off of that and say, how has his work impacted the future of plant breeding?
Yeah, I think it's safe to say that Mendel's work and findings has triggered a flurry of research into the genetics of plants, but not only plants, also animals and humans. And when you want to make progress in plant breeding, you have to know the inheritance of the genes, of the traits that you're studying that are your target traits for breeding. Without Mendel, without having that knowledge, I think breeders would just be making random crosses, and they would be selecting for the biggest ears or the nicest plants without really knowing what they are doing. And of course, we know that it's not so simple as Mendel tackled it. Mendel had the luck that most of the characteristics that he was studying are regulated by one gene only. We now know that in many plants, for many characteristics, those are regulated by more than one gene. But nonetheless, his work triggered all that research into the diverse modes of inheritance in all those other species, and that allowed breeders to make that progress. And without that, I think new varieties, it would take much longer before new varieties, valuable, new, better varieties, would come to the market. And that is something that we cannot afford at this point in time, where we have the massive, unprecedented challenges that we're being faced with climate change, growing population, changing diet, an enormous push towards more sustainability. We need to do more with less acres and so on. And we cannot afford to slow down variety development. We need to make sure that we get drought-tolerant varieties as soon as possible, that we get resistant varieties as soon as possible. So without the knowledge that Mendel provided us, all that progress would be much slower, in my opinion.
Yeah, I agree.
I think it certainly would have taken much longer and might have manifested a little differently. I think it also, the key thing about Mendel is how foundational it is to how we think about things. And some calculations I do for some simple parts of my breeding work, it would be things that Mendel himself could relate to, and it helps us look, how can we devise strategies to get where we're going in plant breeding? How much resources do we need? How can we be more efficient? When I have molecular tools, a genetic marker to be able to track something, that's a Mendelian locus. But also as a foundational, there's the things that became more complex that really set us up to go to the next level. So as I'm making crosses between peppers that are spicy or not spicy, Mendel was working with those contrasting states. For me, not spicy is, zero heat, zero pungency. Spicy is, well, the question everyone has is, how hot is it? So you have this quantitative, continuous variation, this whole spectrum of how that character can exist. And so as I'm tackling that, that's definitely building off of what Mendel was working on. And within that is many genes interacting. And it turns out if I were to discover each of those individually, each of those is working in Mendelian fashion. It's still just a gene, but it's how it works with the other genes. That it's really, Mendel really set us up to take those next steps into everything we do in plant breeding, how everything looks, how we conceptualize a lot of things. It was huge.
Yeah, I totally agree with Michael. I think and the foundation Marcel laid out, and as I think about how plant breeding has evolved, and I think of kind of in three phases, if you will, but all of it is based upon really some of the principles that Mendel put in place. I mean, we've started, and for many years we were in sort of a phenotypic mode. In other words, we would take plants, make crosses, and see what the products of those crosses were, and observe them and note their phenotypes. We then, with the advent of all the modern biology tools and molecular tools, we're able to now, instead of taking everything to a greenhouse or to a field, use tools, molecular markers, other tools to say, actually, we can predict this trait will be there based on the molecular composition. I mean, he didn't know what a gene was, but basically it's the genetic composition. So that transition from a phenotypic selection to a molecular selection. And then now we're evolving to what Michael touched on, which is what are the combination of traits, very much predictive genomic kind of modeling. And so with that, we've moved from where we're taking everything basically at the whole plant level, now to the molecular level at the, at the genetic basis, now to a computer based. And so you're going from the field to the laboratory to a computer to actually do the breeding to do the breeding selections you're doing. But all of it is based upon some of the fundamentals that Mendel laid forth around traits, around quantification, around scale. One of the things he taught us was scale matters. And so you need the statistical significance to actually make the kinds of conclusions he made. But as I think about the way modern breeding programs work and this transition that we're making and we are making from a phenotypic to a molecular to a predictive modeling kind of breeding programs, it's all based upon the principles that Mendel laid out.
Fantastic points. Thank you guys so much. Unfortunately, that is all the time we have today. Again, thank you, John, Marcel, and Michael for joining me, and I hope everyone in our audience took home some fun knowledge today. If you like this topic, make sure to tune in next week where my colleague, Ashley Robinson, is talking about breeding techniques. See you